These are holocarpic and monocentric. Capillita are lacking in the sporangium. Presence of naked sporangia. Presence of cellulosic walls around spores. And isogamous sexual reproduction. One of the mixed amoebae in the cluster becomes larger by engulfing other mixed amoebae. Karyogamy occurs inside mixed amoebae, now called the macrocyst. The macrocyst undergoes meiosis to release mixed amoeba. Cellular slime molds are also called communal slime molds. A life cycle of the cellular slime mold, asexual as well as sexual. Here we can see pseudoplasmodium, which is giving rise to the fruiting body, and free amoebae is released, which grows and multiplies, fuses, diploidy takes place, meiosis is taking place and giving rise to a macrocyst. The sporangium are naked. Cellular slime molds have the characteristic features of both plants and animals. The examples are Dictyostelium and Polyspondylum. Protozoan protists. Since the protozoan protists have more characteristic features of animals, they have been discussed under the phylum Protozoa in the kingdom Animalia. Biological classification of the kingdom Monera. The groups under Monera are Eubacteria, Archaebacteria, Actinomycetes, Mycoplasma, Spirochetes, Rickettsia, Chlamydia and Cyanobacteria. Coming to the characteristic features of monerans, they are unicellular as well as multicellular. They are prokaryotes without a nuclear membrane, nucleolus, chromatin and histone proteins. A true nucleus is lacking as well. The nucleoid or genophore or the incipient nucleus or the prochromosome is composed of naked DNA and RNA and non-histone proteins. The membrane-bound organelles are absent. Cyclosis is absent and ribosomes are of 70S type. What is cyclosis? It is the streaming movement of cytoplasm within a living cell without the deformation of the external cell membrane. The cell wall is made up of peptidoglycan with the exceptions of Archaebacteria and Mycoplasma. The respiratory enzymes are found associated with the plasma membrane. The reproduction is of asexual kind. The cell division is amitotic, that is, the nucleus divides without the division of chromosomes and the cytoplasm divides as well. There is no spindle formation in the cell division. Monerans play a role in our daily lives. They are chiefly decomposers concerned in the cycling rotation of metabolites like carbon, hydrogen, nitrogen and sulfur in nature. They have an active role in symbiosis as well. Some forms show symbiotic relationships such as rhizobium in the roots of leguminous plants or nostoc and anabena in the coralloid roots of cycas, which fixes free atmospheric nitrogen into nitrates, and Escherichia coli, which inhabits the colon of the human intestine and helps in the synthesis of vitamin B. A problem appeared in the AIIMS Entrance Examination 2006. In prokaryotes, chromatophores are specialized granules responsible for coloration of cells, structure responsible for organizing the shape of the organism, inclusion bodies lying free inside the cells for carrying out various metabolic activities, internal membrane systems that may become extensive and complex in photosynthetic bacteria. And the correct answer is that chromatophores are internal membrane systems that may become extensive and complex in the photosynthetic bacteria. Archaebacteria. They are the primitive group of bacteria and are the oldest of living fossils. They are prokaryotes and unicellular. They are found in very harsh conditions, that is, at the bottom of the sea or in volcanic vents. The archaebacteria lacks muramic acid and peptidoglycan. The lipids contain phytanyl side groups, which are branch chain lipids, decrease membrane fluidity and increase tolerance to extremes of heat as well as low pH. The ribosomal proteins are unusually acidic. The archaebacteria are of three types. They are the methanogens, the halophiles and the thermoacidophils. The methanogens, as you can see, are obligate anaerobic form of gram-negative bacteria that produces methane gas from carbon dioxide or formic acid. It's found in the rumen, which is the first part of the stomach of cattle, as well as in marshy areas. The cell wall contains proteins, polysaccharides, example methanosarcina or pseudomurine. Muramic acid and peptidoglycan is absent. Halophiles. The halophiles are the gram-negative obligate anaerobic forms associated with coccoid forms of bacteria. They are very fond of salts, hence they can grow well in culture medium containing as high as 25 to 35% of salt. In the presence of sunlight, they develop a purple pigment which is known as bacteriorhodopsin to produce ATP. However, this particular ATP is not used by them to synthesize their own food. Thermoacidophils. They are the aerobic forms of gram-negative bacteria found in hot sulfur springs, that is, they are found in extremely acidic conditions with very high temperatures. They are facultative anaerobes and are chemosynthetic in nature, the examples being thermoplasma and thermoproteus. They can survive in areas with temperatures as high as 230 degrees Fahrenheit and a pH well below 2. In the AIPMT exam 2008, there was a problem. Thermococcus, Methanococcus and Methanobacterium exemplify bacteria whose DNA is relaxed or positively supercoiled but which have a cytoskeleton as well as mitochondria, bacteria that contains a cytoskeleton and ribosome, archaebacteria that contains protein homologous to eukaryotic core histones, and archaebacteria that lacks any histones resembling those found in eukaryotes but whose DNA is negatively supercoiled. And the correct answer is that archaebacteria that contains proteins homologous to eukaryotic core histones. Next problem is, in the light of recent classification of living organisms into three domains of life, that is bacteria, archaea and eukarya, which one of the following statements is true about the archaea? The archaea completely differs from both prokaryotes and eukaryotes. Archaea completely differs from prokaryotes. Archaea resembles eukarya in all respects, or archaea have some novel features that are absent in other prokaryotes and eukaryotes. And the correct answer is that archaea have some novel features that are absent in other prokaryotes and eukaryotes. Eubacteria. It is one of the most important groups under the Monera. Anton von Leeuwenhoek discovered bacteria and termed them as wild animalcules. Ehrenberg coined the term bacteria. Nageli placed the bacteria under schizomycetes and called them fission fungi. Louis Pasteur proposed the germ theory of disease. He discovered bacteria causing chicken cholera, invented anti-rabies vaccine. He coined the term microorganism. He is considered as the father of modern biology and also the father of sterilization techniques. C. Delort used the term microbe instead of animalcules. Habitat of eubacteria. They are cosmopolitan in distribution, that is found everywhere in air, water, soil, plant and animal. The size varies, ranging from 0.1 to 1.5 micrometer in diameter and 2 to 10 micrometer in length. Smallest is the rod-shaped Dialister pneumocyntis, that is 0.5 to 0.3 micrometer long, present in the nasopharynx of man during the early stages of influenza. Spirillum laidlaw, Epulepsicum fischelsoni, which is 600 micrometer by 80 micrometer, and Thiomargarita ramibiensis, which is 750 micrometer, are among the largest of unicellular bacteria. This is an eubacterial cell with its structural organization. Filamentous bacterium Beggiata mirabilis is the largest bacterium, size ranging from 16 to 45 micrometer diameter and several centimeters long. Cohn classified eubacteria as 5 types: the micrococci, the diplococci, the streptococci, the staphylococci and the Sachinae. Coming to the coccus. Micrococci, example is Micrococcus luteus. Diplococci, example Diplococcus pneumoniae. Streptococci, example Streptococcus lactis. Staphylococci, example Staphylococcus aureus. Sachinae, example Sachina. Bacillus, they are rod-shaped, cigarette-like with round or blunt ends. Most common shapes are motile or non-motile. They may occur in the following shapes. Monobacillus occurs singly, Diplobacillus occurring in groups of 2, Streptobacilli found in a chain Streptobacillus. If the chains have larger area of contact with each other, it forms trichomes. Example is Beggiatoa. Cells form unicellular hyphae in some bacteria, example Streptomyces. Cells line sideways like matchstick form arrangement called as palisade, example Corynebacterium diphtheriae. Cohn classified eubacteria as 5 types: the micrococci, the diplococci, the streptococci, the staphylococci and the Sachinae. Coming to the cocci, micrococci or coccus, they occur singly. Example is Micrococcus luteus. Diplococci found in pairs, Diplococcus pneumoniae. Streptococci, the cells remains attached to form a chain, Streptococcus lactis. Staphylococci, irregular bundles of cells or grape-like clusters, Staphylococcus aureus. Sachinae, they are three-dimensional geometrical figures like cubes. Example is Sarcina. Coming to the Bacillus, or plural is bacilli. They are rod-shaped, cigarette-like with round or blunt ends. Most common shapes are motile or non-motile. They may occur in the following shapes: monobacillus, that is occurring singly. Diplobacillus occurs in groups of 2. Streptobacilli, they are found in chains, and if the chains have larger area of contact with each other, it forms trichomes. Example, Beggiatoa. Cells form unicellular hyphae in some bacteria. They are the Streptomyces. And the cells line sideways like matchstick-like arrangement known as palisade are the Corynebacterium diphtheriae. Bacilli, monobacillus, singly occurring. Diplobacillus in group of 2. Streptobacilli in chains. Beggiatoa forming trichome-like structure. Streptomyces hyphae-like. Corynebacterium diphtheriae showing an arrangement of matchstick-like. Vibrio, bacteria with less than one complete twist and resembling a comma in appearance, causing cholera in man. Spirilla, coiled form of bacteria exhibiting twists with one or more turns, giving a spiral appearance. Stalked bacterium, example Colobacter. Budding bacterium, example Rhodomicrobium. Flagella is one of the main important structures of bacteria. Presence of single filament of globular protein called flagellin in the flagella. The parts of the flagellum are basal body, which is very complex, comprises of 4 rings L, P, S and M in gram-negative and 2 rings S and M in gram-positive bacteria. Hook is made up of different protein subunits. Filament, the longest part of the flagellum. The protein molecules are arranged in a spiral manner and around 20 nanometers long, consisting of 8 vertical rows of flagellin. Coming to the flagellal arrangement. Now this is an arrangement of a gram-negative bacteria. L, P, MS and C rings are seen. This is the rod. This is the outer membrane. This is the peptidoglycan layer. This is the cytoplasmic membrane. And this is the hook, which is also made up of proteins, and filament is the longest part. The flagellar structures. They are of the following kinds: monotrichous, that is a single flagella at one end. Lophotrichous, single flagella at either ends. Amphitrichous, number of flagella 2 or more on one end, and peritrichous, several flagella arising from the whole body. Now, as described earlier, monotrichous, a single flagellum usually at one pole. Amphitrichous, single flagellum at both ends of the organism. Lophotrichous, 2 or more flagella at one or both poles. Peritrichous, flagella over the entire surface. Pilus and fimbriae are used in the case of conjugation. Gram staining technique. Bacteria is identified as gram-positive or gram-negative bacteria. The stain was introduced by Christian Gram in 1884, and That is how we can differentiate between a gram-positive and a gram-negative bacteria. Bacteria is initially stained with crystal violet or gentian violet. All bacteria, irrespective of gram-positive or gram-negative, will stain bluish or bluish purple. Then it is treated with a modin, that is 0.5% of iodine solution. It will be fixed due to the formation of a CVI complex. Then it is destained with alcohol or acetone. The group which does not stain come out to be gram-positive, and the group which stains is the gram-negative bacteria. However, the gram-negative becomes colorless on treating with destaining agent, since it has thin cell walls and has lipids in the cell walls. What is the difference between the gram-positive bacteria and gram-negative bacteria? The thickness of the cell wall is 150 to 200 angstroms in a gram-positive bacteria, while it is only 75 to 120 angstroms in a gram-negative bacteria. Cell wall is single-layered, smooth and in contact with the cell membrane in a gram-positive bacteria, whereas in a gram-negative bacteria we find the cell wall wavy and in contact with cell membrane only at a few places. Since it is wavy, it cannot have contact with the cell membrane at all places, and it is of two-layered. The outer wall is made up of lipopolysaccharide and inner is of peptidoglycan. Lipid contents in the cell wall of a gram-positive bacteria is 2 to 4%, while it is very high in the gram-negative bacteria, that is 20 to 30%. The muramic acid content is 70 to 90% in a gram-positive bacteria, while that in a gram-negative bacteria is 5 to 20%. Amino sugars are 10 to 20% in gram-positive bacteria and 2 to 8% in gram-negative bacteria. Mesosome is present, mesosome absent in a gram-negative bacteria. Pili very rare in a gram-positive bacteria, very common in a gram-negative bacteria. Now the gram-positive bacteria occurs in the shape of coccus and are non-capsulated, while the gram-negative bacteria are mostly bacilli, capsulated, and flagellation is common. Teichoic acid is present and treated. Teichoid acid is absent. Now, what does the teichoic acid do? It actually is a polysaccharide and it gives rigidity to the cell wall. So, in other words, the cell wall of gram-positive bacteria is more rigid than that of a gram-negative bacteria. Gram-positive bacteria produces toxins, and they in the form of exotoxins, and the gram-negative bacteria produce endotoxins. The gram-positive bacteria is sensitive to penicillin, while the gram-negative bacteria is non-sensitive to penicillin, that is, it does not respond to the group of penicillin drugs. Diamine opimatic acid is absent in gram-positive bacteria and absent in gram-negative bacteria as well. Porins absent in the cell wall, porins are present in the cell wall. Few pathogenic forms are there, and gram-negative bacteria has a huge number of pathogenic forms. On treating with lysozyme, protoplast is formed. On treating with lysozyme, spheroplast is formed. The examples of gram-positive bacteria are Bacillus, Clostridium, Staphylococcus, etc. Gram-negative bacteria are Escherichia coli, Xanthomonas and Vibrio. Difference between gram-positive and gram-negative bacteria. Diagrammatically, we can see that the gram-positive bacteria has mesosomes which are enfoldings of the plasma membrane, while it is absent in the gram-negative bacteria. While the gram-negative bacteria has pili which are used during conjugation is absent in the gram-positive bacteria. The gram-negative bacteria also has porin proteins which are absent in the gram-positive bacteria. There was a question in the AIIMS entrance examination 2006. Assertion: gram-negative bacteria do not retain the stain when washed with alcohol. The reason for this is the outer face of the outer membrane of the gram-negative bacteria contains liposaccharides, a part of which is integrated into membrane lipids. State if both assertion and reason are true and the reason is the correct explanation of the assertion. If both assertion and reason are true but the reason is not the correct explanation of the assertion. If assertion is true statement but reason is false. If both assertion and reason are false statement. The correct answer is both assertion and reason are true and the reason is the correct explanation of the assertion. We know that the cell wall of the gram-negative bacteria is very thin and it contains lipids. Therefore, the reason is the correct explanation of the assertion. Glycocalyx. In bacteria, glycocalyx is a sugar coat of polysaccharides that project from cellular surface. It serves to protect the bacteria by creating capsules. Like Streptococcus pneumoniae adheres to the lungs with the help of glycocalyx. This glycocalyx, as you can see, is made up of proteins, polysaccharides and lipids. The cell envelope consists of outermost glycocalyx, middle cell wall and the innermost cell membrane. The glycocalyx protects the cell and also helps in the adhesion. It is represented by either slime layer or capsules. The slime layer is composed of dextran, dextrine and levain, while capsule is made up of polysaccharides and dextroglutamic acid. The slime layer protects the cells from loss of water and nutrients. Capsule provides gummy and sticky characters to the cell. Coming to the cell wall, which is made up of peptidoglycan or murine or mucopeptide. The glycan portion forms the backbone of peptidoglycan, which is composed of alternating units of N-acetylmuramic acid and N-acetylglucosamine, which are joined together by beta-1,4 linkages. The teichoic acids are acidic polymers consisting of carbohydrates, phosphates and alcohol. Porins are the channels for the entry of low molecular weight substances. And the gram-negative bacteria have lipopolysaccharides that act as surface antigen in cell wall. Here we can see the cell wall of a gram-negative bacteria. The peptidoglycan layer, the cytoplasmic membrane, the outer membrane. What are mesosomes? As we have already seen, it is present in the gram-positive bacteria, wherein the plasma membrane shows enfoldings into the cell. There are two types of mesosomes: the central mesosomes, which holds the nucleoid and helps in separating the nucleoid and the septa formation. Another is the peripheral mesosome, which helps in storing respiratory enzymes, for example cytochrome oxidase. Here you can see a mesosome in a bacterial cell, which is an enfolding of the plasma membrane. These are the other organelles of a bacterial cell. Plasmid. It was discovered by Lederberg and Hayes. The extra chromosomal genetic element found in bacteria besides the normal chromosomal DNA are known as plasmids. They are circular and have extra genes. They are capable of autonomous replication in the cytoplasm of the bacterial cell. Now there are various types of plasmids. The sex plasmids, which carry sex fertility factor responsible for the transfer of genetic material during conjugation. The R plasmid confers resistance to antibiotics having a resistance factor, which is known as RTF. The Col plasmid produces special proteins called colicins or bacteriocin to kill other bacteria. And degradative plasmid, which decompose hydrocarbons in petroleum. Here we can see the bacterial chromosome within a bacterial cell, and this is the extra chromosomal material within this bacterial cell, which is known as the plasmid. Coming to nutrition in bacteria. As we can see that nutrition in bacteria are of two types, the autotrophic and the heterotrophic. The autotrophic bacteria are self-feeders, and heterotrophic, as we all know, are divided into three categories, that is the saprophytic, the symbiotic and the parasitic. Autotrophic bacteria are again of two types, the photoautotrophic, which responds to light, and the chemoautotrophic, which responds to chemicals. The photoautotrophic nutrition. It is divided into three types. The photolithoautotrophic, they are the purple sulfur bacteria. They use inorganic sulfur compounds as electron and hydrogen donors. Example, Thiospirillum. Next photolithotropic bacteria are the green sulfur bacteria, which use hydrogen sulfide as electron and hydrogen source. Example, Chlorobium. And lastly are the photolithoorganotropic bacteria, which are purple non-sulfur bacteria, use non-sulfur aliphatic organic compounds as electron and hydrogen donors. Examples are Rhodospirillum. The chemoautotrophic bacteria are the nitrifying bacteria, the Nitromonas and Nitrobacter, the iron bacteria, that is the Ferrobacillus, the hydrogen bacteria, that is the Hydrogenomonas, the sulfur bacteria, which are the Bigiota and Thiobacillus, and the methane bacteria, that is the Methanomonas. Respiration. Coming to the respiration in bacteria, the bacteria has been divided into following types based on their mode of respiration. Firstly, the obligate aerobes. They can perform only aerobically. Example is Bacillus subtilis. Obligate anaerobes, which can perform only anaerobic respiration. Example is Clostridium botulinum. Facultative aerobes are the anaerobic forms but can live in the presence of oxygen. Example is Chlorobium. Facultative anaerobes are the aerobic forms but can live anaerobically. Example is Pseudomonas. Then comes the aerotolerant anaerobes. These bacteria continue to perform anaerobic respiration even in the presence of oxygen. That is Lactobacillus. The aerotolerant aerobes are the aerobic bacteria which continue to perform aerobic respiration even in the absence of free oxygen by using oxygen of the oxidized salts. Examples are the denitrifying bacteria. Reproduction in bacteria. Binary fission is a common method of reproduction during favorable conditions wherein the bacterial chromosomes replicate, giving two circular chromosomes. It is also known as the theta model since it looks like the Greek letter theta during a stage of chromosomal replication. This mechanism was given by Kahn, hence known as the Kahn's model as well. In this, the bacterial population is counted by 2 to the power n, where n is the number of generations. What is the process of binary fission? The cell elongates and the DNA is replicated. The cell wall and plasma membrane begins to divide. The cross wall forms completely around the divided DNA and the cells separate. Here we can see that the cell elongates, the DNA replicates, and there are two daughter chromosomes. There is a wall in between the two cells, and here are the two separate daughter chromosomes. Endospore formation. It was discovered by Kohn in hay bacteria, that is the Bacillus subtilis. Thick-walled, highly dehydrated and resistant spores formed under adverse conditions. The commonly formed in genera like Bacillus and Clostridium. The endospore formation is more means of perennation than reproduction, that is, the spore persists for a longer period of time under adverse conditions, and on return of favorable conditions they give new offsprings. The cortex and cytoplasm contains calcium and dipicolinic acid, which prevents the protoplasm to coagulate at higher temperatures. Here is a structure of endospore. This is a spore cell, the exosporium, the spore coat, the outer membrane, the cortex and the intermembrane. The endospore formation. See here, we can see the end of vegetative growth. Then there is a asymmetric septation. A septa is formed between the two. This is smaller and this is bigger, that is asymmetric. Then what happens? There is an endocytosis or engulfment of the daughter protoplast. Here the daughter protoplast is engulfed within the cell. Synthesis of peptidoglycan containing the cortex. Then there is an assembly of proteinaceous coat, and finally there is a lysis of, that is breaking of mother cell, and the spore is released. Genetic recombination. The true sexual reproduction is absent in bacteria, but genetic recombination is there, which comprises of the three methods: the transformation, the conjugation and the transduction. They are known as the parasexual method as well, because they do not involve syngamy and meiosis. Transformation. The donor and recipient do not come in contact with each other. A bacterium picks up small DNA fragments of killed bacteria from the surroundings with the help of membrane receptors. Bacteria that are capable of being transformed are known as competent